Okay, folks, so, and apologies for the movie, this is the TSI follow-up. So you guys, you just shared with me the TSI stands for triple sugar iron. What are the three sugars? Glucose, sucrose, and lactose. The same three sugars that we ran in the PR test, right? Okay, and you just told me that to inoculate, you use your needle, stab the butt to force your microbes to grow anaerobically, and then you streak the slant so they can grow aerobically. Okay, so folks, we know we have three sugars. What does the I stand for? Iron, okay. And you guys, the iron is in there so that we can safely determine whether our microbes make hydrogen sulfide. Now you guys, hydrogen sulfide, it's toxic. Remember how we described in metabolism how it inhibits cytochromes? It'll shut down our electron transport chains. And it smells obnoxious to it. It looks like rotten eggs. And I'm thinking that's probably natural selection, right, for us to avoid areas that have hydrogen sulfide. So we don't want you detecting hydrogen sulfide by taking the cap off the tube and taking a big whiff, right? Because it's toxic. So the microbiologists figured out if you add iron salts to the TSI medium, the iron will combine with hydrogen sulfide to make a black solid precipitate. And this black solid, you guys, is called iron sulfide. And we can actually see it as black in the agar. Now, the problem is with our Citrobacter, when I checked yesterday, only like one team out of 6 had any black. So can you guys just hold up your Citrobacter? Oh, good, awesome. Oh my gosh, you guys. Over the night, they read the lab man. Oh, yay! Okay, all right, you guys, yay. Because I was afraid yesterday that we were only going to have one team that had positive hydrogen sulfide. You guys over here, any and you guys, truly, this is nothing you did wrong. I still can't figure this out, why some of them are making hydrogen sulfide and others aren't. When we read in our Bergey's manual, which is kind of almost like our Bible, it says that hydrogen sulfide is strain specific. So you can have a range from like 20 to 70% of the strains will make hydrogen sulfide. But what puzzles me is this is all the same strain, and why in some tubes it's making hydrogen sulfide and others it's not. I still haven't figured that out, you guys. I'll have to talk to an expert about it. Okay, so are there teams where your Citrobacter didn't make any hydrogen sulfide, meaning there's no black? Okay, so you guys, what's going to be really important is you ask your colleagues, can you see their tubes? Okay, so that you can cartoon their tubes so you feel confident that you can recognize that black precipitate. Okay, so you guys, this gets tricky, however. So folks, if on the lab exam, we ask you, what is that black solid? What is the black solid? Iron sulfide, right? And it tells you your microbe made what? Hydrogen sulfide. Okay, so what gets confusing, somebody say the black is hydrogen sulfide. Is that correct? No. The black is iron sulfide. But it tells you what? Your microbe made hydrogen sulfide, right? A colorless toxic gas. So folks, another thing we could ask you about hydrogen sulfide production is how would your microbe make hydrogen sulfide? And there's two ways. One is through anaerobic respiration. If they use sulfates as alternate terminal electron acceptors, the sulfates get reduced to hydrogen sulfide. Okay? A second way they can make hydrogen sulfide is by taking amino acids like cysteine that have a sulfhydryl group, an SH group, and as they're tearing the cysteine apart, they're releasing the sulfhydryl groups as hydrogen sulfide. So that's a second way your microbes could make hydrogen sulfide. Okay? All right. Okay, folks, so we got the hydrogen sulfide out of the way. That's kind of the most complicated one. Now let's return, folks, to sugar fermentation. Okay? Do you know which pH indicator is in TSI? Do you know which pH indicator is in TSI? Phenol red. Good job, you guys. And folks, what color is phenol red at acidic pH? Yellow, good. What color is phenol red at alkaline pH? Red, yeah, that deep red fuchsia color. Okay, and folks, I've got a poster up here. But I think for the movie, I'm going to use this one because I think it's going to be easier to see. Okay, so you guys, this gets a little bit tricky. So for sugar fermentation, we score the slant and then we score the butt. So you guys, if we have, let's take this first tube here, we've got a yellow slant and a yellow butt. So yellow phenol red tells us is the agar alkaline or acidic? Acidic. Okay, so you guys, we score the slant and then the butt. So you guys, do we have an acidic or alkaline slant? Acidic. So we have an A, acidic slant, and what about the butt? It's also acidic. So we have A slant, A butt. Now what does that tell us? Now this is where it gets complicated, you guys. So if the butt is acidic, that tells you glucose was fermented. If the slant is acidic, that tells you lactose or sucrose or both were fermented. So folks, an example of a lab exam question. If I show you this TSI, first of all, help me score it. What's the slant? A, acidic. What's the butt? A, acidic, right. So you guys, was glucose fermented? Yes. Was lactose or sucrose or both fermented? Yes. Good. And in addition, folks, we can tell whether molecular hydrogen or carbon dioxide was produced during fermentation by looking for gas production. It might be subtle. It might be just cracks or bubbles in the agar. But is it subtle here? No, right? And this makes sense, you guys, because we know at least 2 of the sugars were fermented. And that means if this microbe makes gas, it's going to make quite a bit of gas. Yeah? So you guys, help me out. Does your E. coli, let's look at your E. coli slants, folks. Okay, your TSIs. So you guys, how would you score your E. coli? A slant, and what's the butt? A, right? So we know from the PR sugars, you guys, that E. coli ferments all 3 sugars. Does that explain why such a massive amount of gas was produced, right? And what happened is that massive amount of CO2 and molecular hydrogen was made. What happened to your slants? They were blown apart, right? Just like a little hand grenade. I came in yesterday, you guys, and vented them again. I used a sterile swab to push the agar away from the lips of the tube because they were resealing. Yeah, and some of them were even sticky because the agar was getting pushed out, so I sprayed them with disinfectant. But definitely, guys, presume they're contaminated even on the outside. So make sure you throw your gloves in the biohazard bin, okay? All right. So now, folks, this one's a little bit trickier, and I know it's hard to see here in color. But this second tube, you guys, the butt is yellow and the slant is red. So let's score it. So is the slant acidic or alkaline? Alkaline, good. And if the butt is yellow, the butt is acidic. Okay, so you guys, this microbe only fermented glucose. Okay, so remember, an acidic yellow butt tells you glucose was fermented. If there's no yellow in the slant, that means neither lactose nor sucrose was fermented. Okay, we all right with that? Okay. This next one, you guys, is a tough one. This is one we have to memorize. I don't think I'd ask you on this exam to memorize this one, you guys, but let's do the simple part first. What's the black? Iron sulfide. Iron sulfide, and it tells you your microbe made what? Hydrogen sulfide. Name 2 ways your microbe can make hydrogen sulfide. Anaerobic respiration using what is terminal electron acceptors? Sulfates. Good. And what's the second way your microbes can make hydrogen sulfide? Tearing apart the sulfhydryl-containing amino acids like cysteine. Awesome, you guys. All right. Now, folks, when you have this much hydrogen sulfide, we just have to memorize, if we could see through the iron sulfide, that would be a yellow butt. So a yellow butt tells us what? It's acidic, and it tells us glucose was fermented. The slant, you guys, is red, so that's alkaline, and that tells us what? Neither lactose nor sucrose was fermented. Good job, folks. And what about this last one? It's all red, right? So alkaline slant, alkaline butt. What's that telling you? No fermentation of the sugars, right? So, for example, folks, remember we did Pseudomonas as a demo in the PR sugars. Remember how it didn't ferment any sugar? This could be a Pseudomonas, right? Doesn't ferment any sugar. Good. And what does it do? It can tear apart amino acids. It doesn't make hydrogen sulfide, but it can tear apart amino acids as an alternate carbon and energy source. And we know when amino acids are torn apart, what's the effect on pH? Yeah, pH goes up. We have an alkaline reaction, so we're going to get the nice deep red of the phenol red. Does that make sense, you guys? That's a lot of information. I always had to really work on that one to remember that. That's a lot of information. Okay, so you guys help me out with your E. coli. So you already told me you scored it as an acid slant, an acid butt, molecular hydrogen or CO2 was produced, because, right, there's so much gas. Was hydrogen sulfide produced? No, right? Because there's no black iron sulfide. Good. Folks, what about your Citrobacter? How would you score the slant? Okay, acid slant, an acid butt, folks. So what does that tell you about the sugars fermented? Glucose for sure, and sucrose and or lactose. Good, excellent. And folks, those of you that had black, did your Citrobacter make hydrogen sulfide? Yes, good, good. And again, those of you that have Citrobacter that didn't make any hydrogen sulfide, could you look at your colleagues, right? Because if we have it on the lab exam, I want to make sure that you get it, okay, without, being misled. All right. Gosh, you guys, I think that is it. So let me close the movie down, folks, and then we'll ask for any questions. And then my thought was, you guys, after you have a chance to diagram your results, right, because you're going to turn in your results with your Lab Exam 2 packets, that those of you that want to, this is going to be totally optional, you guys. Those of you that want to, what we would do is just go through your lab study guide packet, and I would just use it as a trigger to ask you quiz questions. So, but you guys, we know it can't be comprehensive, right? We can't in like an hour go through 8 weeks. Right, but it'll just be, I'll just go through the study guide packet and see if something is like, oh man, I love to ask something about this question, right? And then I can quiz you on it. But it's totally optional, folks. Just consider the rest of the lab an open lab, okay? So if you feel like you can make better use of your time, like studying in the library, don't feel like you have to stay here. Okay, but you guys do be really careful, right? Make sure that. Take all the tape off your tubes, barely loosen the caps, put them in the kill area. Remember, you guys, your gloves go in the biohazard bags bin, not in the regular trash. Make sure, you guys, that you wash your hands and spray your bench before you leave, okay? All right. Oh, yeah, and I'm so sorry. Let me just save this really quick.